Hello everyone, in this video I'm going to be talking about the Krebs cycle. Now this video is a sequence of videos that I've been doing on cellular respiration, where we're looking at aerobic respiration and then finally anaerobic. Now in the previous part, so in part 1, we looked at glycolysis and the link reaction, and we said that in those two, or at the end of those two stages rather, in the process of respiration, what we had done is produce pyruvate. We had taken an organic compound, namely, let's say, glucose, and we had gone through glycolysis to make it pyruvate, and this is with oxygen. So we were first talking about the aerobic route of respiration, and we said that glycolysis primarily happens in the cytoplasm, and then for the link reaction, which is kind of like the link into the Krebs cycle, that happens in the matrix of the mitochondria. And that's where this story really picks up, because at the end of the link reaction, what we have is acetyl-CoA. So we produce an intermediate compound called acetyl-CoA. We've had carbon dioxide being produced in a process known as oxidative decarboxylation, and we've produced some NADH, or reduced NAD, a reduced coenzyme along the way. So, Krebs cycle. You can see top right of the screen, we've got a picture of the man himself. It's important to acknowledge his work because Hans Krebs, born and raised in Germany, has done so much for the field of science. In 1932, he discovered the Urea Cycle, and that was essentially reactions in animal organisms, producing urea from ammonia. He actually fled to England in 1933, where the Nazis took power, and Krebs, being Jewish, was denied academic employment. He eventually landed a job at the University of Sheffield in 1937, where he made these infamous discoveries, and in 1953 he actually got the Nobel Prize in medicine. It's just a little bit there about the man behind the process, because it's such a well-known, famous cycle that we're going to look at. So this here is the Krebs cycle, also known as the Citric Acid Cycle. That's one thing to note. So what we'll do, in the same as I did in part 1, at the end of the cycle I'll talk about the net results, or the net gain of all the different compounds that we've produced. We'll put those together with the ones from glycolysis and link reaction, and we'll get a final, or a total amount of products before we enter the final stage of respiration, oxidative phosphorylation. So I'm going to draw a Krebs cycle out, and we'll start with the acetyl-CoA. So let's just draw in, fact I'll just draw this in green very, very quickly, just for those that want it. The actual chemical structure, so we have H3C here, we've got a C with a double bond O, S, and a CoA part there. So we've got the chemical structure, if you like, of acetyl-CoA. Now acetyl-CoA is a 2-carbon molecule. I'm going to look very carefully at the carbons. I'm going to circle them in red. Acetyl-CoA, what we've produced from the link reaction, is our starting point, because that will feed into the production of a 6-carbon molecule. Now that 6-carbon molecule is called citrate. Now we've gone from a 2-carbon to a 6-carbon, so clearly there must be a 4-carbon somewhere feeding into this, and there is, in this 4-carbon here feeding in, is a chemical known as oxaloacetate. So what I'm going to do in this video is essentially build up the Krebs cycle, and then I'm going to explain the important chemicals that come off it. So acetyl-CoA, our intermediate compound, joins with oxaloacetate to form a 6-carbon compound, citrate. And that's step 1. Now I'm going to do this in quite some detail. So we're going to say that is step 1. In certain textbooks they skip between, they talk about the 6 carbon going to a 5 carbon, then to a 4, and then that's where they kind of leave it. But I'm going to go into a little bit more depth to explain exactly what those individual steps are. So step 1 is producing citrate. That 6 carbon molecule, citrate, goes on and through a process which you don't need to necessarily worry about for this video, produces another 6 carbon compound, and that's step 2. I'm more doing this just for completeness sake. Now at step 3, we change that 6 carbon into a 5 carbon, so the C means carbon, a 5 carbon compound. And that is step 3, and at step 3 here, that is where something interesting happens. That is where we get NADH being formed, NADH, and an H plus. And we also get, I'll draw this in another colour, CO2 being given off. So what we can see is that we've got a decarboxylation here. So CO2 is removed and lost as a sort of waste gas, hence why we've gone from a 6 carbon to a 5 carbon, because one of our carbons has been lost in the CO2. But we've also created some NADH, or reduced NAD. So that means that the 6 carbon must have lost or donated the hydrogens to NAD to form NADH. So in part 1 I talked about the coenzyme NAD that can be reduced or given hydrogens to help oxidize another chemical. So in this case, the 6 carbon compound has been oxidized to form the 5 carbon compound. It's lost the hydrogen, and that hydrogen has been given to NAD to form NADH. And in fact, a second hydrogen is removed, and that's why we've got an H+. So right now we've got a 5 carbon compound. We then have that 5 carbon compound producing a 4 carbon compound. And again, at this point here, when we go from 5 to 4, we get NADH, an H being released, and another CO2. So we've lost the carbon through the formation of CO2, so we're giving out CO2 in this process as such. And again, we've oxidized the 5 carbon compound to 4 carbon compound because we've produced NADH. So NAD has been reduced to allow us to do this step. Now this is where things get a little bit interesting. So at the 4 carbon compound, that then becomes another 4 carbon compound, and that here, if we were to go back and say that this is step 4, then the one we're looking at now is step 5. And at step 5 in this Krebs cycle, what we have is something called GTP. Being released. Now GTP fuels the conversion of ADP, adenosine diphosphate, to ATP, adenosine triphosphate, in what we call substrate level phosphorylation. So I'll put that here. So basically we get what's called substrate level phosphorylation. And GTP, I'll just make a reminder instead of note here. So at substrate level phosphorylation, basically we're getting ATP from that. So GTP allows us to create ATP, if you like. Now, if we pick up after step 5 with the 4 carbon compound that we have 2 more stages of producing 4 carbon compounds, like so. So you can see why some textbooks, if we say this is step 6, this would be step 7, and this one here would be step 8. You can see why some books would shorten it and just have 6 carbon, 5 carbon, 4 carbon, and then 4 carbon adding to the 2, the acetyl-CoA, to go back to 6. So it kind of keeps it a bit shorter, but I deliberately want to show you all of the steps in the Krebs cycle. So let's pick up with step 6. At step 6, what we have being released at this point here is something called FADH2. Now, there is a coenzyme called NAD, and we've talked about that because that can then form NADH. But there is another coenzyme called FAD, and that can form FADH. And in this case, what we've actually done is create FADH2. So in step 6, what we've had is one 4-carbon compound donate or lose 2 hydrogens, so it's become oxidized. And in doing so, it stayed as still with 4 carbons, but it's become a different chemical compound. So this 4-carbon at step 6 has changed into another chemical, which also is a 4-carbon in step 6. But in doing so, it's been able to lose hydrogen, donate it to FAD, and reduce FAD to produce FADH2. Nothing really happens in step 7, and it's step 8, the last step 8, that there is where we get a final NADH being produced and an extra H+. So we've oxidized the 4-carbon compound there to produce NADH. So you can see that Krebs cycle is ultimately a series of redox reactions, just oxidations and reactions. That's basically it. And what we're doing is using acetyl-CoA, the intermediate compound from the link reaction, we're combining that with oxaloacetate and forming citrate. And in this cycle, we're producing a number of things as a result. Now you've got to remember that this, like I said before with the link reaction, this cycle is done twice, and we'll explain why. And we'll finish this video by looking at ultimately the net gain of chemicals. So if we shrink the screen a little bit here and move this just up a bit so I can make this fan light. So if we look, so at the end, so this is what I've done in part 1, I've looked at the end at what we've produced. We have produced 2 lots of CO2. We have 3 lots of NADH made. We have 1 lot of GTP, which will go to make ATP. So we'll just call it ATP for now. So 1 lot of ATP and 1 lot of FADH2. That's what we've had produced from this cycle, this Krebs cycle. But remember, we said that in glycolysis we end up producing 2 pyruvates for every 1 glucose. So we had 2 lots of the link reaction because there were two molecules of pyruvate going through. So equally, in Krebs cycle, if both pyruvates go through the link reaction to produce acetyl-CoA, technically we would have 2 lots of the Krebs cycle, because we have 2 pyruvates that would ultimately enter the Krebs cycle. So all of this would need to be times by 2. So at the end of Krebs cycle, for both pyruvates from glycolysis, what we've got is 4 CO2, 6 lots of NADH, 2 lots of ATP, and 2 lots of FADH2. So just to finish, if we add everything up from part 1 and part 2, the 2 videos that I've done so far, if we think about what we've made in glycolysis, the link reaction and Krebs as a complete total, then what we've got at this stage in respiration, aerobic respiration, is 4 lots of CO2 from this cycle, plus we produced 1 CO2 in the link reaction, but because we have 2 lots of the link reaction because there are 2 pyruvates, in total we've produced 6 CO2 up to this point in time. We've got 6 NADH from Kreb cycle alone, but there were another 4 in total made prior to this. So in total what we've got is 10 NADH. In the first stage of glycolysis we used 2 ATP but made 4, so we've got a net gain of 2 plus the 2 now, so we have got a total of 4 ATP, and we have there the 2 FADH2. So here in blue is our sort of net gain. If we consider glycolysis, the link reaction and Krebs cycle, this is what we've produced up until now. The CO2 is released as waste. The ATP we're going to use as energy. Now the NAD, let's just make that a bit clearer there with the 10, because I drew the box over it, so 10 NADH. The NADH and the FADH2 will then feed into the final stage of cellular respiration, which is called oxidative phosphorylation. And it's there where we use these coenzymes to produce vast quantities of ATP, which is the purpose of respiration. It's obtaining energy from the breakdown of an organic compound. So the last stage in respiration that we're going to look at is the stage where we use these products, if you like, of Kreb cycle to get us lots of ATP. Okay, I hope all that helps.